Justin Sinclair is a research fellow at Nickham Health Research Institute, Western Sydney University, and also a lecturer here at our Sydney campus. Justin coordinates the Australian Medicinal Cannabis Research and Education Collaboration at Nickham. For the last 3 years, he's also served on United in Compassion's Scientific Advisory Board, Australia's leading medicinal cannabis patient advocacy group, in a pro bono capacity. Justin is currently a co-investigator on several clinical research projects in development around medicinal cannabis and spends much of his time lecturing to medicinal practitioners, nurses, politicians and other allied professionals about the therapeutic applications of the cannabis genus. He's published on the topics of cannabis and the endocannabinoid system, pain management and herb-drug interactions in peer-reviewed journals, and is a member of the Society of Cannabis Clinicians and International Cannabinoid Research Society. So much there. I just wanted to give you all a grounding of who Justin is in this specialisation. So from now, Justin, I'm going to pass it over to you to introduce yourself any further and get cracking. So I'll talk to you all later.
Fantastic. Thanks very much, Raleigh. So good evening, everyone, and thanks very much for the kind introduction. And one thing I just wanted to start out with was starting this talk by making something quite clear from the outset, which was the difference between recreational cannabis and medicinal cannabis. And it's really important to understand that the two are very, very different things. So one's grown illegally or illicitly in many instances, and in some cases poorly, utilizing different things like insecticides or fungicides or growth regulating agents, whereas medicinal cannabis is actually grown to incredibly high quality standards, making it much safer for therapeutic use and medicinal applications with a more standardized chemical profile for reproducible therapeutic results. So we have to compare apples and apples when it comes to this discussion. And one thing as we go through this that I ask you to keep in mind is that most of the evidence for cannabis and its perceived harm, increased risk of psychosis, toxicity, dependence and addiction has actually been based on recreational evidence, and in the vast majority of the literature, with almost none that's actually being done on purely medicinal quality assured product. So with that being said, I do have quite a bit to cover today. Raleigh's indeed right, so we'll jump straight in. I'm going to take you on a brief journey about the cannabis plant, and we'll look at its botany and morphology, its chemistry, but not before I actually brief you on an entire neuromodulatory system that we'd actually know nothing about if it wasn't for research into the cannabis genus. So without further ado, let's get started. So on this slide is just a, in the interest of transparency, there's just a list of my affiliations and disclosures. So one thing I just wanted to note was that I've not been paid nor received payment from any medicinal cannabis cultivators or manufacturers either here in Australia or overseas. And all of the talks that I've done, this is actually my 43rd talk in 2.5 years that I've given to industry, doctors, nurses, patients and carers and students. I've received no money for this, so it's purely because of education and trying to get the good word out. So with that being said, and with the time allocation, I want you to strap in, make sure your trays are stowed in the upright position, and here we go. So the evolutionary development of the endocannabinoid system has actually taken place over millions and millions of years and across a great range of biological organisms. We actually find evidence of the cannabinoid receptor genes that have been found in Siona intestinalis, the sea squirt, which is a deuterostomian invertebrate, suggesting that these receptors actually developed in this class around 500 million years ago. So it's not just unique to us. Further investigation, particularly along animal models, has found that those cannabinoid receptors were also distributed fairly widely throughout the animal kingdom and has been documented in different groups such as fish, amphibians, mammals, leeches, sea urchins, and mollusks. And research into how the phytochemicals in cannabis interacted with human physiology was actually quite a slow evolution also. It took place in a coordinated fashion over many decades. So this academic investigation into cannabis has been largely impeded due to inconsistent cannabis plant samples, prohibitive laws hampering availability and research, which actually started in 1937, and a poor understanding of the chemical structure of the phytocannabinoids, that is, the cannabinoids that come from plants. So with advances in techniques in analytical separation in the 60s, Gayoni and Meshulam first elucidated delta-9 tetrahydrocannabinol in 1964, and that's the main psychoactive component attributed to cannabis. And then 3 years later, they actually chemically synthesized THC, which made it much more available to researchers worldwide. Now, some 20 years later, it took, in 1984, studies then elucidated that cannabidimetic agents, that is, those agents that work via a similar mechanism, were found to inhibit cyclic AMP accumulation in neuronal cells, and that this process was done via a receptor-mediated cellular response. 4 years later, in 1988, Meshulam and colleagues actually demonstrated that THC was stereospecific. For those of you that don't know what that means, it basically means that it requires a specific site of action for binding, and so they started going on the hunt for the receptors. And later that same year, the first cannabinoid receptor was actually identified by Devane, and 2 years later it was structurally expressed and defined by Matsuda. Now, by this time, research interest started to grow internationally and at a fairly rapid pace. And in 92, the next bit of the puzzle, which was what endogenous ligands, what endogenous substances do we produce and manufacture by us, the organism, to actually bind with these receptors? And that was discovered. So based on what researchers already knew about phytocannabinoids and that they were highly lipophilic or highly fat-loving, researchers hypothesized that these endogenous ligands would also be lipid-derived. And that was proven correct again by Devane and his research group, who identified anandamide, known chemically as N-arachidonyl ethanolamine. So a year later, in 93, the second cannabinoid receptor was isolated and has since been updated. And in 95, one of the main endocannabinoids, which is 2-arachidonyl glycerol, or what we just shortened to 2-AG, was discovered by Meshulam and Sugura. So lastly, and actually not discussed on this slide, is the evolving research into the enzymes involved in the production or synthesis and degradation catabolism of the endocannabinoids, which largely became a target of pharmaceutical interest in the late 1990s. So basically, the endocannabinoid system represents an incredibly wide-ranging neuromodulatory system which is involved in the regulation of many facets of homeostasis. Indeed, it may actually define how homeostasis is maintained within the body. And we see cannabinoid receptors widely expressed in many cells and tissues throughout us. So essentially, we break down the endocannabinoid system into three major components. We've got the cannabinoid receptors, the CB1 and CB2 receptors, although others are being posed to belong to this group, which are distributed right throughout the various organs and tissues in the body. We've got the endogenous ligands, that is the endocannabinoids like anandamide and 2-AG, that interact with these receptors. And then we've got enzymes that are involved in the synthesis or degradation of those ligands. Now, of key clinical interest is that any individual variability, that is variability of the individual person, or genetic polymorphic modifications to any one of those three components can potentially change or modulate or modify the normal functioning of the entire endocannabinoid system and the body systems that it regulates. So which highlights, I think, the importance of taking a patient-centered approach to the use of cannabis and other cannabimetic agents in patient care. So let's have a look at the cannabinoid receptors first. And these cannabinoid receptors belong to the family of G-protein coupled receptors, what we shorten to GPCRs. And they're expressed in abundance in the central nervous system, where they far exceed those present for the neurotransmitters that they modulate. This will come in a little bit, you'll see how important this is a little bit later. But it's not all about the cannabinoid receptors, the CB1 and CB2 receptors, as you can see, with many different receptor types expressing endocannabinoid binding, with examples being the GPR55, GPR119, again, G-protein coupled, the transient receptor potential vanilloid 1 receptor, the TRPV1, and the PPARs, the peroxisome proliferator activator receptors, alpha and gamma subtypes. Now, the interesting thing about these PPARs is that these are nuclear receptor proteins, very, very important for things like gene expression, cellular differentiation and metabolism via the regulation and expression of genes. So the more we're learning, the more we're actually finding that the endocannabinoid system is a web through which most of our physiological processes are firmly enmeshed within. Now, as most of my students would already know, I'm fairly severely colorblind, so I have it on good authority that purple's easy to see on the diagram. I would appreciate you to let me know if that's not the case so I can seek appropriate retribution with my brother. But the CB1 and CB2 receptors, again, coupled to the G proteins, how they work physiologically is one of three ways. So they either inhibit adenylate cyclase, which is inhibiting the conversion of adenosine triphosphate to cyclic AMP, and hence to cyclic AMP-dependent protein kinases, very important for signaling. Two was the stimulating the MAPK, or the mitogen-activated protein kinases, which are directly involved in various cellular responses based on stimuli such as mitogens or osmotic stress. Pro-inflammatory cytokines are another good example here. And therefore, these assist in regulating various cellular functions such as things like mitosis, gene expression, apoptosis, and cellular differentiation. So what we see here is that stimulation of the MAPK is a direct mechanism by which the cannabinoids have been implied or posited to impact synaptic plasticity and possibly even neural regeneration and growth. And lastly, the modulating the activity of the potassium and calcium ion channels, which assists in the transducing the binding of agonists. So this latter protein-mediated modulation of ion channels includes activating the inwardly rectifying A-type potassium channels or inhibiting the voltage-gated calcium channels. And what this does is modulate the neurotransmitter release of things like noradrenaline, acetylcholine, and glutamate being examples. We'll talk a little bit more about this in a few slides. So the CB1 receptors are distributed fairly extensively throughout the brain and the central nervous system. The high CB1 levels in both the motor regions and sensory regions of the brain are fairly consistent with the important role of the CB1 receptor in things like cognition and motivation. Now, CB1 activation, listen to this, inhibits the neurotransmitter release of GABA, glutamate, dopamine, serotonin, acetylcholine, D-aspartate, and noradrenaline of both the inhibitory and excitatory synapses. So when you think about that, CB1 receptors are primarily involved in the modulation of this neurotransmitter release centrally, but also have roles in causing other things such as coronary and cerebral dilation, neuroprotective signaling, the relief of pain, and they've even been shown in vitro studies to promote neural stem cell differentiation. Now, but CB1 receptors are the same receptors that delta-9 tetrahydrocannabinol, that's THC, from cannabis binds to cause the various psychotropic and therapeutic actions. For example, the amygdala, responsible for emotions, fear, and anxiety. And as such, in certain patients, THC can cause can cause these things as a side effect, particularly in varieties of cannabis that have been bred to have quite high THC concentration. So the cerebellum involved with balance and motor coordination, and not surprisingly, THC can cause this to be impaired also. So the effect of THC on the hypothalamus is thought to be why it increases appetite. Wonderful for cancer patients, HIV, AIDS patients. And it can also impair short-term memory due to acting on the hippocampus. So take that to heart and maybe keep that in mind when it comes to study preparations. Okay. The other thing that's really interesting too is that it alters pain sensitivity by working directly on the spinal cord, particularly an area called the gelatinous region. So a particular clinical interest, though, is there's a sparse expression of these CB1 receptors in the brainstem, which many of you know involves areas that are responsible for cardiopulmonary centers. So this can explain the lack of respiratory depression that you see with cannabis use, but is something that's quite commonly observed in opiate medication overdose. Interestingly, also, the anti-nausea effect of cannabis is believed to directly act on this particular brain structure. So the CB1 receptor itself is also present, but in much lower concentrations in the peripheral neurons and non-neural regions such as the vascular endothelium, the testes, epithelial cells, the eye, and several other peripheral organs, which we'll get to. Now, the CB2 receptor expression, on the other hand, is largely dominated in the peripheral immune cells and tissues, currently considered responsible for the effects that cannabinoids have on immunomodulatory activity. And this is largely thought to be due to the CB2 signaling pathway, which plays a critical role in regulating microglial activities within the CNS. And you know the important protective aspects of the microglia in the central nervous system. So CB2 receptors do not cause any noted psychoactive activity, but do contribute to the local analgesic effects, so very useful for pain. Furthermore, the CB2 receptors demonstrate very important neuroprotective and anti-inflammatory activities, with CB2 expression actually being enhanced by inflammation, which is suggestive that this receptor subtype is involved in the endogenous response to injury or trauma. So recent evidence has also demonstrated the role of CB2 activation in analgesia in both acute and neuropathic models of pain. Now, next step on the brief journey to understanding the endocannabinoid system is the ligands that interact with those receptors we just talked about. So the term endocannabinoid merely represents our body's own endogenous ligands that interact with these cannabinoid receptors to elicit a physiological therapeutic response. And for the most part, two endocannabinoids have seen the lion's share of the research, and we've mentioned them already. That's anandamide and 2-arachidonyl glycerol. So anandamide was the first one that was isolated in 92 and was so called or named after the Sanskrit word ananda, meaning supreme joy or bliss, due to its cannabinomimetic activity. And that's just pertaining to anandamide's ability to interact with the cannabinoid receptors and mimic the psychotropic effect of THC. So as you can see, several other endocannabinoid-like compounds have been identified, and we're still undergoing research into their exact roles within the body. Although palmitoyl ethanolamine is actually being studied by a research group, I believe, up in Queensland at the moment. So here's an example of the enzymatic synthesis and degradation of anandamide. Once again, I apologize for the kind of garish colors. I have had people tell me that it's an affront to the Lord himself, but I apologize for that, but it's literally all I can see. So this catabolism, or more specifically the hydrolysis of anandamide, is mediated primarily by the enzyme fatty acidamide hydrolase, FAAH, which degrades anandamide to arachidonic acid and ethanolamine. So alternatively, though, anandamide can be oxidized by certain cytochrome P450 enzymes, lipoxygenases, and cyclooxygenase 2. So what we see here, this diverse and complex enzymatic mechanism involved in endocannabinoid metabolism, is important for what we call maintaining the resting tone of the entire neuromodulatory system. So this diversity, I think, also highlights that any inherent abundance or deficiency of these enzymes in the individual could have a fairly dramatic effect on the overall functioning of the ECS. So as such, if the enzymes responsible for, say, endocannabinoid degradation were suppressed experimentally, a prolonged therapeutic activity of the endocannabinoids is achieved. And this is demonstrating yet another very promising target for cannabinoid pharmacotherapy. Now, 2-AG, on the other hand, appears to actually be the true ligand for the cannabinoid receptors rather than anandamide, and it's the key endocannabinoid involved in something we call retrograde signaling within the brain, and I'll show you that in a couple of slides. Now, 2-AG can be produced by several other different metabolic pathways, more so than anandamide, which does show or highlight its importance. And current research actually suggests that 2-AG is far more abundant in the central nervous system than anandamide, hence why we go back to that idea that it is the true ligand. So the catabolism of 2-AG mimics that largely of anandamide via the oxidative processes, again from the cytochrome P450 enzymes, lipoxygenases, and cyclooxygenase 2, although as you can see there, the specific enzymes involved in synthesis and degradation are different, with monoacylglycerol lipase, or MAGL, being an example. Now, interestingly, one thing you might not have noticed is that unlike anandamide, I'll just cycle back fairly quickly, But what you notice there about anandamide is it's only a partial agonist for the CB1 and CB2 receptor, but it's a full agonist for the TRPV1, very important for inflammation. Whereas when we go back and look at 2AG, we see that it is actually a full agonist for both the CB1 and CB2 receptor. Now, as promised, I said this is the really interesting stuff. So within the nervous system, these endocannabinoids are currently believed to be synthesized on demand. So they're not stored in vesicles like most neurotransmitters. So they're synthesized on demand from stimuli such as a response to injury, ischemia, or stress. And it's generally done in an activity-dependent manner. So I apologize for the arrow. The arrow is actually should be flipped all the way around, because what we're wanting to see is this tracking of the catabolism of things, of phospholipid membrane components on the postsynaptic neuron. So down the bottom, where that postsynaptic neuron is, these endocannabinoids, mainly 2AG, are manufactured on demand from phospholipid membrane components, and then they exert their physiological effect by traveling back across the synaptic cleft and binding to cannabinoid receptors on the presynaptic neuron. So what this does is inhibit the various inhibitory or excitatory neurotransmitters that are present there. So this action is what we call retrograde synaptic flow, and it's the primary activity noted by Meshulam of the endocannabinoids. So pretty fascinating stuff. I'm sure you're aware of most of what you've learned when it comes to transmission of nerve impulses, and probably haven't heard of anything quite this specialized. Now, due to this anatomical distribution of the endocannabinoid system through the brain, spinal cord, the enteric and peripheral nervous systems, it plays a pivotal role in regulating a broad list of physiological processes that are involved with homeostasis. So listed there, and I don't need to talk too much to them, but regulation of stress and emotions, nociception, that is pain, cardiovascular respiratory function, appetite regulation, thermogenesis, psychomotor behavior. So I'm sure as you read through that list and look at the extensive distribution of the endocannabinoid system throughout our physiology and anatomy, it is fairly clear to me and many other researchers that this is the main neuromodulatory mechanism we have for the response of homeostasis. Now that we've touched on a little bit of the anatomy, what we're going to do is look at some physiology. So with this ubiquitous distribution that we see of the ECS throughout the central and peripheral nervous system, we believe the ECS plays a very meaningful role in neurological pathophysiology, particularly epilepsy. Now, early studies have demonstrated defects within the endocannabinoid system in epilepsy patients, with one study on patients with newly diagnosed temporal lobe epilepsy actually exhibited significantly lower levels of anandamide in their cerebrospinal fluid when tested against contrast to healthy controls. So it's very interesting. Further supporting that hypothesis that the ECS can play a role in seizure inhibition in epilepsy was a study done on resected tissue following surgery for epilepsy, whereby these epileptic patients demonstrated lower levels of CB1 receptor messenger RNA expression in the glutamatergic terminals of the dentate gyrus, in contrast to post-mortem controls of non-epileptic subjects. Interestingly, same study, there was a reduced expression of DAGL alpha was found in epilepsy patients as well, which, as I'm sure you probably won't remember, but it was the enzyme responsible for 2AG synthesis postsynaptically. So we're really starting to see dysfunction of the endocannabinoid system consistently rear its head when it comes to epilepsy. But it's not just epilepsy. Reduced levels of anandamide have also been identified in the cerebrospinal fluid of patients with chronic migraine, with studies subsequently done showing the gene encoding for the CB1 receptor, which is known as the CNR1 gene, being linked to chromosomal regions that have been identified and marked or triggered by, or associated with, is a better word, sorry, migraine. So variations in CNR1 gene expression have been shown to predispose to higher risk for migraine development in earlier research. So not surprisingly, a clinical endocannabinoid deficiency has been posited by a researcher known as Ethan Russo, and as being critical in our understanding of the pathophysiological understanding of things like migraine, and also certain functional pain conditions such as irritable bowel syndrome and also fibromyalgia. And research, particularly into fibromyalgia, is currently ongoing. But it's not all about deficiency. Excess levels of endocannabinoids have also been noted in the literature. And here we see point 2 here is of great interest, I find. This is previously it was largely believed to be the domain of endorphins, natural opiates, runner's high, as they say. So producing our own endocannabinoids as a response to exercise gives new meaning, I think, to the term runner's high, and also changes our previous chemical understanding of things such as reward-seeking behaviors. So it wasn't all endorphin-based. Now we understand that the endocannabinoids, particularly anandamide, have a place here too. Now, increased levels of anandamide have been found in both the plasma and cerebrospinal fluid of schizophrenic patients, in contrast to healthy controls. And furthermore, single nucleotide polymorphisms, SNPs, have also been described for the CNR2 gene, which codes for the CB2 receptor, with researchers actually suggesting that susceptibility for schizophrenia may be increased by a genetically predetermined decrease in the functioning of the CB2 receptor. And more research is being conducted on this in the United States and Japan at the moment. But it's not all about the nervous system. Let's now move on to the cardiovascular system, as the endocannabinoid system does play a fairly important regulatory role in CVS physiology and the development or progression of common cardiovascular disorders. Now, studies have shown that in cases of heart failure, that endocannabinoids are produced by activated monocytes, which in turn contribute to the development of things like hypotension and negative inotropic activity, so reducing the force of contraction of the heart. Now, further research demonstrated that under pathophysiological conditions in test subjects, animals, cardiac myocytes and vascular smooth muscle cells can also generate their own endocannabinoids, which can then interact with the CB1 receptor and can actually cause problems like reactive oxygen species or advanced glycation end product accumulation, which could then contribute to cell death and injury by accumulating in the spaces. So of interest here is that an upregulation of CB1 receptors and an increase in endocannabinoid expression were noted in the myocardium of these experimental models. So interestingly, there was also a study done by Rajesh and colleagues that suggests that the CB1 receptor in the cardiovascular system promotes cardiac dysfunction, oxidative stress, inflammation, and fibrosis when looked at in diabetic cardiomyopathy. Conversely, just like a light switch, the CB2 receptor involvement in the cardiovascular system actually appears to be protective, with activation decreasing pro-inflammatory and fibrotic responses and initiating protective effects in cardiac myocytes. So CB2 activation reduces immune cell chemotaxis, inflammatory cell adhesion and cellular activation, and appears to be behind the protective effects in preclinical models for things like myocardial infarction, stroke, restenosis injury or reperfusion, and atherosclerosis. So look, a lot more research needs to be done to actually look at the influence of the endocannabinoid system over the cardiovascular, and more research needs to be required. But there's a fairly clear dysfunction in the endocannabinoid system is present, particularly this CB1 overactivity in various cardiovascular diseases, and that CB1 antagonists, i.e. blockers, and CB2 agonists are of a fairly great clinical interest, I think, from a pharmacotherapeutic point of view to these specific cardiovascular disorders and conditions. Now, what about the immune system, I hear you say? Well, there's a growing research base is showing that anandamide actually has been shown to inhibit immune function by reducing the production of things like pro-inflammatory cytokines, whereas 2-AG, acting through the CB2 receptor, may inhibit the migratory activities of certain immune cells by modulating various mechanisms. Now, of particular interest is that studies have shown anandamide reduced things like interleukin-6 and interleukin-8 in human monocytes, whilst also suppressing the release of interleukin-2, tumor necrosis factor alpha, interferon gamma, and activated T lymphocytes. And all of these, the activated T lymphocytes was via the CB2 receptor. So this is perhaps why many autoimmune and neurodegenerative patients are utilizing cannabis, in many cases illegally, but they're finding some therapeutic relief. And now a snapshot of the ECS physiology in the gut. Most endocannabinoid activity in the gut is actually mediated by the CB1 receptor under normal physiological conditions, with things like mesenteric vasodilation, motility stimulation, acid suppression, and fluid secretion being the main examples. So the list that you've got there is the current homeostatic mechanisms involving the GIT, which have been outlined. But what's interesting is that with a growing research base demonstrating the connection between a balanced intestinal microbiota and a normalized immune system, GIT system, and neurological functioning, I'm sure it's of no surprise to those listening in the natural medicine field to learn that CB1 expression on enterocytes is actually regulated by the enteric microbiota. Isn't that incredible? With CB1 activation increasing epithelial permeability by reduced expression of tight gap junction proteins, which could then permit bacterial translocation and potential metabolic endotoxemia as sequelae. So blocking CB1 expression, therefore, may help normalize intestinal barrier function. This could be a new treatment for leaky gut and has also been investigated for reducing obesity. Conversely, the CB2 activation within the gut can exert a normalizing activity in instances by, some examples of increasing gastric motility observed in specific GIT conditions. And still in the GIT, the liver has a sparse population of cannabinoid receptors, but CB1 receptors are found in hepatocytes and vascular endothelial cells and stellate cells, whereas the CB2 largely are found in immune cells and the Kupffer cells in the liver. Now, research suggests that CB1 activation has been associated with vasodilation, leading to things like ascites, increasing fat deposition, and insulin resistance and fibrosis. And not surprisingly, CB2 exerts an anti-inflammatory activity, reduces cytokine production, and has an opposing effect to that CB1 activation, and may be a very new and interesting line of research into conditions affecting the liver, such as cirrhosis and non-alcoholic fatty liver disease. Now, key to understanding the endocannabinoid system is that every individual will express this system with slight difference, with slight variability. Some individuals can express an abundance of receptors, but may not produce a lot of their own endocannabinoids, a seeming endocannabinoid deficiency. Conversely, others may exhibit the opposite presentation. Now, variability in the development and expression of the endocannabinoid system, whether it's those receptor numbers or expression in tissues, the endocannabinoid concentrations, or the enzyme amounts, will all significantly impact the resting and functional tone of this entire neuromodulatory system, which would therefore impact homeostasis. So it may also potentially modify how phytocannabinoids, that is the cannabinoids produced from plants, or synthetic cannabinoids, those synthetically derived in the lab, can interact with the individual. And what excites me most is that the more we're learning about the ECS, it actually seems likely we will obtain a much greater understanding of the etiology and pathogenesis of many medical conditions, which currently we just don't have answers for. And that's something, when you look at autoimmune conditions, over 80 autoimmune conditions, and we don't know what cause hardly any of them. So with this brief summary now done on the ECS, let's now have a look at the phytocannabinoids. Let's have a look at the plants, or the major plant, and how they are manufactured. All these chemicals are manufactured by the plant. We'll look at some potential dosage forms, some herb-drug interactions, and then I'll finish up with a recent survey that we conducted looking at cannabis and endometriosis. Okay, so now that you understand how the ECS works a little better, let's have a look at some of the botany of the cannabis family. So the Cannabaceae family is a relatively small family of flowering plants. It's only got around 11 genera and approximately 170 different species. And currently, and for many years, it's been hotly debated that there are largely two major species within cannabis. That's Cannabis sativa and Cannabis indica. And also a third is considered there for you on the slide called Cannabis ruderalis, although many authors and breeders believe it to be phytochemically inferior. more to the point, with so much interbreeding of the different cannabis species and selective plant breeding programs in different countries around the world, producing hybrids and different varieties or chemovars, cultivars, if you will, of what we commonly refer to as strains, which is rather incorrect, but cultivar or chemovar is probably more correct. It's actually a bit of a moot point to discuss what species might be useful for a certain medical condition. What we really need to do is actually analyze the individual phytochemistry of the plant to know what it might be useful for. And that's what I pose, particularly from a scientific standpoint. So as you notice for medicine there, we typically want the female unfertilized inflorescences. Inflorescence, just that fancy term I'm sure some of you remember for many tiny flowers. But I'll talk more about that soon. I've got some photos that I will show you. Now here's a map that presents the current present-day ranges of the cannabis gene pool. And this was taken from a book called Cannabis Evolution and Ethnobotany by Mark Merlin and Robert Clark. Now, firstly, and perhaps most worthy of note, is that in the zones that are capable of supporting plant life, what do you find? Cannabis is present. And second is that cannabis is seen on every major inhabited continent because our ancestors took it with them wherever they went because it was actually highly valuable. They needed it as a staple, revered it for its versatility and hardiness. They used it for a food and fiber, not just as a medicine or for entheogenic purposes, spiritual and ritualistic purposes. So now that we've gone through this, let's just have a quick look at some of the different morphological characteristics of the cannabis genus, the different shapes of the plant and parts of the plant, so that you can see what parts are used medicinally. Okay, so here we have two leaves that look quite similar. They both share the classic tapered acuminate apex that you can see there, compound leaf structure, serrated leaf margin, but the length and width of the individual florets there and the leaflets, and the chemical characteristics in these two cannabis plants are markedly different. So whether broad or narrow leaf species or subspecies, both will share medicinal and industrial applications. Now, a lot of you might not know this, but the leaves are generally regarded as a waste product in cannabis cultivation circles. However, there's a lot of anecdotal and case report evidence of patients juicing them, and this was actually found in I think it was the Taylor case in Penrith Court not too long ago, where a dad was charged with growing, I think, over 100 plants, but he was juicing the leaves and the flowers for his two daughters that suffered from inflammatory bowel disease, and putting them in some cases into remission. And that was largely because of the higher concentrations of tetrahydrocannabinolic acid, which is the precursor for THC. So it's actually the acid form of THC. It's non-psychoactive and does have anti-inflammatory properties. So it's not just about the flowers or the trichomes. So here on this slide on the left you can see a male cannabis plant, and the stamens are comprised of the filament upon which the anther sits, of course. I'm sure you remember your botany, and that produces the pollen. So you can see here in figure 4 the old male cannabis plant produces a lot of pollen, and it travels a great distance on the wind. And for this reason, much like Catholic school growing up for me, it's generally kept isolated from the female plants, except for when breeding was desired. They've actually even found the pollen from plants grown in Morocco on plants that were growing in Ireland, some 3,000 kilometers away. So it can certainly be taken long distances on the wind. So if that female plant gets pollinated, it's going to produce large amounts of seed, and that's going to detract from its ability to produce larger amounts of phytochemicals for medicine, and will generally negatively impact the potency of the medicine. So as such, the standard for producing medicinal strains or chemovars of cannabis is to ensure that only unfertilized female plants, or what are commonly called sensimilla, is cultivated, and then generally cuttings or clones or tissue culture is taken from those unfertilized mother plants so that they have the identical genetic makeup and will assist in maintaining a consistent quality of the phytochemicals required for medicine. So you'll notice there's this kind of hazy whitish coating on the female plant on the right, and that's certainly not a mold or a fungus, but actually another very interesting morphological characteristic of the plant known as trichomes. Now on this slide you can see two female cannabis plants. We've got on the left, you can see that feathery looking female reproductive part known as the stigma, and that's upon which the male pollen lands when fertilization is meant to occur. And you can also see the glandular trichomes, which are these little hairs, magnified more on the right for you, which is where some of the phytochemicals of medicinal interest are stored within the plant, particularly the cannabinoids and the terpenes or the terpenoids. So as the plant reaches maturity or senescence, these clear trichomes will become more opaque and almost milky under magnification, and that's actually showing the cultivator that the time for harvest is fast approaching. Now hashish, which many of you may have heard of, is a resinous extract composed of just these glandular trichomes and represents a very strong end product that's been used in ritual, ceremony, and medicine by humans for many thousands of years. It's essentially just purified cannabinoids and terpenes, so much less is required to be used as a medicine in comparison to just the dried plant material. Now here on this slide, figure 8, you can see these compressed glandular trichomes forming solid form hashish. And if you were to view that under a microscope, you just see amazing massive compressed trichomes. Now hashish is largely produced two major ways, either shaking or rubbing. Now shaking requires the use of incredibly fine material through which the raw cannabis product is bashed and sieved, and just the trichomes break off and fall through the netting and are collected. Now rubbed hashish is much easier to produce, and we see that generally in cultures such as India and Jamaica, where they involve simply rubbing the flowers of the many living cannabis plants, and after about 20 to 30 minutes your hands become coated in that sticky resin, as you can see there in figure 9, which is effectively pure hash. So it's then just scraped off with a knife and rolled into balls or pressed into forms. And as I said, this is this is largely known as charas from India, and is predominantly used in India and the Himalayan regions. Now here we've got two finished trimmed inflorescences showing the glandular trichomes. So the specific strain that you see here is Baba Kush, and that was actually named for its paying homage, if you will, to its ancestry being a broadleaf drug variety from the Hindu Kush region of Afghanistan. Very, very high producer of resin and high in THC. Figure 11 is another rich resin strain known as Afghani Kush, and again, interestingly, high in THC and well known for sedation and pain relief. Why I put these pictures here was to show that both of these are Kushes and they share similar parentage and varietal line, but you'll notice that they both express quite different phenotypic differences in color and trichome abundance. And that's why we go back to that idea of it's not about the species. The plant is so unique and able to adapt to many different environments, which will all change its phenotypic and morphological expression and chemical expression, of course, as well. Now, something that many of you might not know is that cannabis sativa and cannabis indica are known to be affected by something called photoperiodism. And this is defined as basically the developmental response or physiological reaction of the plant to the length of the daylight cycle. Now, most normal plants, as I'm sure many of you know, go into flower in the warmer spring and summer months. But cannabis doesn't belong to that club. So during spring and summer months, where sunlight exposure is approximately 15 to 18 hours a day, these plants will actually undergo vegetative growth, or veg growth as it's called in the trade. So during that phase they maximize the growth of leaves to pump out as much carbohydrate as they can through photosynthesis, which I'm sure you all remember from your year 9 biology class. So this process provides the fuel to increase the growth and vitality of the plant. It can really start growing and getting healthy. So there's another cannabis species you remember me talking about earlier, which was Cannabis ruderalis. And this is actually known as an auto-flowering variety, meaning that it goes into flower after a certain period of time. It's built into the genes, and it's not actually affected by photoperiodism. And due to that fact, it's largely unwanted in cannabis breeding programs. So here's another picture showing the vegetative growth pattern, but much smaller plants here under indoor lighting conditions. So various spectrums of photosynthetic photon flux density lighting is actually used in the various stages due to the spectrum outputs that are required. So whether you're in veg or whether you're in flower, and some of the examples might be things like high pressure sodium lights or metal halide lights. And because cannabis cultivation indoors costs a lot of money to run these lights and cooling systems, LEDs are being explored a lot more now because they use far less energy. Actually, in the United States alone, 1% of the entire energy grid is given to growing cannabis, which is a fairly significant signature and footprint, isn't it? Not very environmentally friendly. Whereas in Australia, we've got all this amazing sun. We can grow it in greenhouses. So as the sunlight wanes and reduces in length in autumn and winter, dropping to around 10 to 12 hours a day, it starts the process of flowering, which is obviously the main structure that we're interested in medicinally. So this is why cannabis is suited to both outdoor growing and indoor growing, as it's just a matter of light exposure to actually change the plant from the veg state to the flowering state. So you can actually get 3 to 4 grow cycles in a year. Now these handsome specimens, I'm talking about these beautiful plants, of course, look at them, vital, flourishing, beautiful and green, are Cannabis sativa subspecies sativa, known as hemp, and that's grown at a top secret cultivation site. But I stand about 179 centimeters tall, which makes these beauties in the vicinity of around 12 to 13 feet, and there's still about another 4 to 5 weeks of growth before they'll be ready to yield. So another foot or two they could potentially put on. So most indica and Afghan varietals are much smaller and more robust, and that should make sense to you because it's very cold where they usually grow, and if they were to grow large like these beautiful samples, which we typically see more in temperate climates and tropical climates, they would simply freeze. And so the cannabis plant is a perfect example of adaptation to its environment. Okay, so now that we've touched on a little bit of botany and morphology, we'll look at some of the phytochemistry within cannabis and how it interacts with endocannabinoid receptors and not only cannabinoids, but we'll look at another group known as the terpenes, which work via different mechanisms of action. So you've all probably heard of the psychoactive cannabinoid delta-9-THC and maybe about cannabidiol or CBD, which is gaining a great deal of media attention because of its anticonvulsant activity in assisting things like epilepsy. But there are numerous other cannabinoids and other phytochemical classes within the cannabis plant that are also of clinical interest. Now, I always put this slide in, and it's kind of disappointing because I can't see the looks on your faces, but I'm sure it's probably similar to the look of most of my classes when I teach pharmacognosy. So things are about to get a little bit more technical, and I'll try and keep them as pain-free as possible. But let's move on to something maybe that looks a little bit less like the love child of a chemistry textbook and a street map of Sydney. And for our purposes, this is far more manageable. Now, cannabinoids are a class of terpenophenolic compounds that occur in the cannabis plant. And you can see why they're called terpenophenolic on this diagram. As in the top right-hand corner, you've got levotolic acid, which represents the phenolic moiety, and on the left is geranyl pyrophosphate, which represents the terpene moiety. Now, these come together to form cannabigerolic acid, or CBGA, which is the precursor for all other major cannabinoid acids. So it's in this acid form, which is largely carboxylated, meaning that it just has a carbon dioxide molecule or a carboxyl group that I've highlighted in blue for you. So if you went tiptoeing through the tulips of a cannabis plantation and picking flowers and eating them, the cannabinoids exist in this acidic form and you're not going to get high off it because it won't bind with the correct CB1 receptors and it won't cross the blood-brain barrier in that polar form. So far there have been over 144 different cannabinoids classified, but this is largely dependent on the variety of cannabis being tested as they all demonstrate unique cannabinoid profiles. And these are all subject to change based on the temperature, sunlight exposure, elevation, water supply, soil pH, and nutrient availability in the soil. There's so many things that can actually change the expression of the chemistry in the plant. So in the fresh plant, it's these acidic forms that exist, as seen in the rectangle that you can see here at the top of the slide. So not their end products, which we predominantly use as medicines, such as delta-9-THC or CBD. So once we dry and cure the plant material or expose it to heat, such as in smoking or baking edible goods, oops, what's happened there? There's been a problem. Of course there has. Let me try and, actually, I won't do that. I will wait for that to move back up.
The PowerPoint again.
I will do that. And we should be back. Are we back, Riley?
Yes. Yeah, you're back to, yeah,
awesome.
you scroll through, yep.
Okay, perfect. I'll scroll through. Sorry for that. I'm not sure.
Thankfully,
Computers
just past the
so meant
just
to make
past
our
the
lives.
last child of a
There we go.
chemical book.
Yeah, there we go. Okay, so so what we were talking about then was just it's the acid form that's produced, and so what we need to do is actually remove that. So we need to actually expose it to a little bit of heat or drying it, such as smoking it or vaping it or baking it, as some of you may have done. And those carboxyl groups that are highlighted in blue will start vibrating at a really high frequency, and they'll snap off, and that releases carbon dioxide and allows the new decarboxylated product that is formed to actually interact with the various cannabinoid receptors and other receptor types. So here we see down the bottom how this decarboxylation process changes the non-psychoactive tetrahydrocannabinolic acid, or THCA, into the psychoactive delta-9 THC, but the same mechanism exists, guys, for the other acid forms in the plant, like cannabidiolic acid, or CBDA, being converted into CBD, cannabidiol, the product that's used for epilepsy management. Okay, so interestingly, THC binds with relatively higher affinity for both CB1 and CB2 receptors in the central nervous system, very similar to that of our own endogenously manufactured anandamide. So this constituent is associated with that feeling of being stoned or psychoactivity, and if given in high isolated dose, can cause things like dysphoria and potential paranoia and anxiety in certain individuals. Now, pharmacologically, THC is a very well-described analgesic in the management of pain related to cancer, muscle spasm, migraine, phantom limb pain, spinal cord injury, damaged nerves, and even has been used post-surgically. It's shown benefit in assisting with muscle spasticity associated with multiple sclerosis, and perhaps is best known as an appetite-promoting agent for cancer and AIDS patients, and also for reducing the nausea and vomiting associated with chemotherapy. Less well known is evidence supporting its use for lowering intraocular pressure in the eye in those suffering from glaucoma, and reducing symptoms associated with Alzheimer's disease. Now, CBD, on the other hand, is a non-psychoactive or non-inebriant cannabinoid. So CBD actually opposes the dysphoric effects of THC, which is why it's a benefit to ensure that the two constituents are present in some type of standardized ratio, depending on the therapeutic condition that's being treated. Unlike THC, CBD has very low affinity for binding to CB1 and CB2 receptors, but rather works via different mechanisms of action, such as being an agonist for the 5-HT1A serotonin receptor, which may explain why it exhibits antidepressant and anxiolytic activity. Due to CBD's strong antioxidant, anti-inflammatory and neuroprotective actions, it holds great interest in the treatment of seizures, epilepsy, Parkinson's and Huntington's disease, multiple sclerosis, and Alzheimer's. So as I'm sure you noticed, the fact that some of these pharmacological actions are highlighted is showing how these actions are shared across these two constituents, albeit in many cases this will be via different mechanisms of action or by interacting with different receptors. But this is just 2 cannabinoids out of the 144 that are being characterized in the cannabis genus. Now, worthy of note within the minor cannabinoids is delta-8 THC, which exhibits less psychoactivity than delta-9. And if you're ever wondering what delta-9 actually means, it's just referring to the position of the double bond in the chain. So it still expresses many of the same useful therapeutic activities of its stronger cousin THC. THC, the tetrahydrocannabivarin, that's a bit of a mouthful, exhibits anticonvulsant activity similar to CBD and cannabidivarin, and is of interest in improving glucose tolerance in type 2 diabetes, which is obviously a pretty big major public health problem at the moment. Now, cannabinol and cannabigerol, or CBN and CBG, share the ability to decrease proliferation of keratinocytes and therefore show some very interesting promise in the skin condition known as psoriasis. Now, whilst a large amount of these actions that are on this slide have only been demonstrated in animal models or in vitro models, it does highlight how much more there is to this plant from a research perspective, and it's not all about the THC and the CBD. So here's a little infographic I designed that lists the actions associated with just 10 of the various major and minor cannabinoids. So there's still so much more we need to learn and understand about this plant, which despite its illegality still has more scientific publications on it across a wide array of scientific disciplines than any other herbal medicine that I've actually investigated in my 20 years of research in this space. So let's now have a completely different look at another class of phytochemical compounds known as terpenes. Now, as I'm sure you remember, the terpene class are also found and held within the glandular trichomes of the cannabis plant. And it's this phytochemical class that's largely responsible for the unique tastes and aromas and smells associated with the different cannabis varieties. So you can think of these as like the essential oils, if you will, of the cannabis plant. But they're not unique to the cannabis plant, but are also commonly and abundantly distributed throughout many different plant families and species, particularly many of our own used in herbal medicine. Now up the top you can see something like limonene, and it's responsible for the smell of many citrus species. But what you might not know is that that little terpene packs quite a therapeutic punch, not only having strong anxiolytic and even antidepressant activity, but also showing evidence for its use in gourd, and has moderate antibacterial activity as well, and has even been shown to produce cell death in breast cancer cell lines. So these antioxidant and anxiolytic actions highlighted in blue are also shared with CBD, but occur via different mechanisms of action. So having multiple phytochemicals within a plant that exhibit similar pharmacological activity increases this therapeutic potential of the medicine, and is something that we naturopaths and herbalists call phytochemical synergy, but they actually have a term for this in cannabis research where it's called the entourage effect. Then we have a look at beta-myrcene. So this monoterpene exhibits very strong muscle relaxant, sedative, anti-inflammatory and analgesic activity, which, as you know, are actions shared by both THC and CBD. And at concentrations of just over 0.5%, beta-myrcene is associated with the well-known couch lock phenomena, which is kind of like, honey, could you please go get me the Tim Tams because you can't move. Now, lastly, we see the alpha-pinene, and that monoterpene exhibits pretty strong bronchodilatory and anti-inflammatory activities as well, which, as you guessed it, are also shared with the other two major cannabinoids. So many other terpenes exist, of course, with over 200 being identified across the different cannabis varieties alone. So as you can see, there exists a great deal of crossover when it comes to just these two phytochemical classes. But with that being said, guys, over 700 different phytochemicals have been characterized across the various cannabis varieties. So the diversity of pharmacological actions and potential benefit for many different health problems, I think, is fairly easy to see. Now, as cannabinoids and other pharmacologically active phytochemicals such as the terpene class within cannabis are highly fat-soluble or lipophilic, the dosage form of how the cannabis is administered is also very clinically relevant, for the individual patient and the condition being treated. So it may actually determine the difference between treatment failure and the amelioration of symptoms, and also other factors such as how long it will impact the patient, its duration of effect, and its potency. So for example, orally ingested THC-rich dosage forms like oils or capsules will undergo biotransformation in the liver to a much stronger form known as 11-hydroxy-THC. But that will take anywhere between 30 to 90 minutes to take effect. But it will provide longer duration of activity, up to 4 to 8 hours. Whereas smoking or vaping, a similar variety, will provide a much quicker onset of action, typically around 1 to 2 minutes, but a much shorter duration of effect, sometimes only 2 to 3 hours. So the correct dosage form is an integral component of ensuring effective medicinal cannabis strategy, and yet another variable that clinicians and patients, largely medical doctors in Australia, of course, because they're the only ones legally allowed to prescribe cannabis at the moment, need to consider. So not all of them have been well studied, however, as you'll see from this slide, yeah, that's not surprising considering its only recent legal status. Now, getting ready to finish up the talk today, I did want to just talk about some of the known interactions that we need to be mindful of. And we do need to take these fairly seriously for drugs that are metabolized by these pathways, as it could increase serum levels and slow down elimination of the drugs. A particular problem when you consider drugs of narrow therapeutic index. Now, other side effects attributed to cannabis, it's important not just to talk about all the benefits, but there are some side effects that have been included in the literature, include tachycardia, hypertension, and short-term effects on cognition, including things like sense of time, sensory perception, attention span, verbal fluency, problem solving, psychomotor control, and reaction time have all been impeded. But obviously, with cessation of use, these go away. Now, in some instances and with large doses, hallucination, anxiety, and dysphoria have been noted in certain individuals, particularly in those high THC containing varieties. So I guess at the end of the day, whilst cannabis is certainly not the panacea that many claim it to be, at least not to me, and it may not work for everyone, as we start to unfold, I think, these mysteries of the endocannabinoid system and its influence over disease and homeostasis and health, I expect that we will see this amazing plant medicine take its rightful place back in the pharmacopoeia or armamentarium once more, and it's utilized more and more as our evidence base grows. Now, just finishing up now, I just wanted to give a quick talk to you about a study that was led by Dr. Mike Armour. He's a postdoc research fellow at NICM with me, and we put together our brains and decided to put this study together where we looked at surveying women with endometriosis and what they used for self-care. So what were the things that they used that weren't prescribed by doctors that they could do themselves or get guidance on doing? So looking at things like heat packs, cold packs, Tai Chi, acupressure, stretching, breathing exercises, meditation, alcohol, all sorts of things that they use, dietary restriction, herbal medicine, and we looked at all of this. And we did this as an online survey using Qualtrics. So we sought through recruitment through social media platforms. We looked at Endometriosis Australia and EndoActive. They were very, very supportive. And we targeted women aged 18 to 45 that were currently appropriately diagnosed via laparoscopy for having endometriosis. So we ran that from October 2017 to December 2017. Only took about 15 minutes and included things like a pelvic pain impact questionnaire, different demographics and self-care usage, etc., etc. So what we ended up finding was 590 women completed the survey, but only 484 completed it enough that we could use it in the analysis. We found that average age of the respondents was 31 years of age, and that most of them, around 77%, lived in urban areas. Self-care use was pretty high over the past 6 months. So 76% of respondents actually used one of the self-care modalities that was described. And those that didn't use it largely didn't because of lack of information, time, or cost. And that last one definitely is an important factor. So the most popular self-care was heat and heat packs, which many women with endometriosis would probably be nodding their heads frantically right now. Rest, of course, something that's just not tolerated in today's modern society, but is very needed for this particular patient group. And breathing exercises or meditation was only around about 47%. So of these three most popular, only heat was rated as giving significant relief from pain. So that was a self-rated effectiveness of 6.5 out of 10. However, the highest self-rated interventions were illegally sourced cannabis at 7.6 out of 10, heat was 6.5, and cannabidiol oil or CBD oil at 6.33. And then following behind closely were the dietary choices at 6.4 and acupressure at around 6.3. So the champion of this was cannabis, and interestingly, illegally grown cannabis. So physical interventions, things like yoga, Pilates, stretching and exercise were actually quite rated low in terms of effectiveness, particularly when it came to pain management. So there was no specific dietary type we found within this survey, whether paleo, vegan, keto, that was more effective than others in terms of reducing symptoms, although some women swore by it. Adverse effects were pretty high amongst those using alcohol, particularly self-reporting hangovers, and exercise as well. Many women actually reported flare-ups being quite significant after their exercise. And most effective self-care for reducing medication use was cannabis, with 56% of the women that used cannabis illicitly having a 50% reduction in their medication. And as many of you might know, the classic medication that's used by many women for endometriosis are the opiates. So very, very significant finding, particularly when we consider the opiate epidemic that is gracing the developed world at the moment. So self-care rates were similar or even higher than women with primary dysmenorrhea, which was previous research that Dr. Mike Armour had done. And self-care is likely to help women fill the gap where their symptoms aren't being controlled by modern medicine. and was a very, very important coping strategy we found that provided this sense of agency and empowerment, which is obviously incredibly important too. So greatest impacts on women were obviously seen through GI function. There was improvement in mood and energy levels as well, and that obviously related to the specific self-care that was chosen and utilized. So whilst cannabis was only used by a very small proportion of women at 17%, it was the highest rated when it came to affecting both reducing symptoms and the need for modern pharmaceutical medication. Diet, again, provided some consistent benefit, but didn't seem to matter what type of diet they chose. And heat was the only modality that was both effective and commonly used. Adverse effects were actually still reported for that. Some women, put it on so hot that it actually would cause blistering, but it was that heat that was needed to actually, almost like a massive counter irritation, to actually foster some type of analgesic and pain management. Exercise was a problem for these women and problematic for some. Generally, the more gentle yoga didn't seem to be as problematic as some of the more aggressive forms of things like Pilates, and that needs to be investigated further. It'd be great to see, some of you new grads that are interested in following up. There's a lot of research that could be done to further this for women suffering from endometriosis. And so the conclusions that we came with this study, which is currently being peer-reviewed, is that self-care does play an incredibly important role in endometriosis. Cannabis, of course, provides significant relief, but we need to do further studies, and that's exactly what I'm going to be focusing my PhD studies on. So based off this, further surveys will be done internationally, and then I'll be looking at conducting a clinical trial to see if we can bring another medicine into the fray to assist women that have this very, very life-changing condition. We do need to look at exercise a little bit closer. Not all exercise is appropriate, and diet, we definitely need to do some more research here and find out are there certain triggers that might make endometriosis symptoms worse. So I think that's about it. I think I've gone a little bit over, and I apologize for that, but I did want to just squeeze in a little bit of the research that we've been doing because it was relevant to cannabis. So were there any questions at all, Riley?
Hi Justin, I just want to say thank you so much for all the information you've passed on to both our students and graduates, and I mean it's just an eye-opener about how much is happening in this space and the potential there to research more and make a difference in so many people's lives. So thank you so much. We do have a few questions. Have you got a few minutes just to answer a couple?
Absolutely, my pleasure.
Okay, great. Well, if anyone else writes in questions, we're probably not going to get to them, but I'll throw these to Justin. The first one I think you'd like to know is, thank goodness you haven't used red or green because I can't differentiate those two. Purple's great. So,
excellent.
Can 2-AG be made from omega-6?
That's a good question. I'm not entirely sure. I do have a research partner that I could put you in contact with that actually does a lot more of the biochemistry work when it comes to the endocannabinoid system. So my email is actually all lowercase j_Sinclair at Western Sydney, all one word, .edu.au. So whoever asked that question, send me an email and I'll follow that up for you. The whole interesting thing about that though is that with the particularly when you start looking at anandamide and the role of arachidonic acid in inflammation pathways and things like that, and actually seeing that a lot of these phospholipid membranes are actually, all the phospholipids themselves and the endocannabinoids are manufactured from those components, I would hesitate that that's probably the case. But instead of giving you flawed information, I'd much rather go to the guru and I can find out an exact answer for you.
Great. This was done at about 620. Mm-hmm. Maximum decarb temps. I'm not sure what that was. Does that make any sense to you?
Yeah. Look, that decarboxylation, the process by which you decarb the products, can affect what actually comes through. So when you're looking at some of the more fancy vaporizers that they use now, such as the Volcano and others, which actually have temperature settings so that you can dial in what temperature so that you can actually pull out various terpenes or stronger amounts of terpenes and less cannabinoids, or if you just really fire that temperature up and you might burn off some of the terpenes and not get them through as well, but you'll maximize the cannabinoids. Generally speaking, for people that are doing oils and all those types of things, it's wiser, I think, to actually decarboxylate the product first and then add it to the oil and sonicate it to actually extract the maximum amount of cannabinoids and terpenes. And typically that's done at about 100 to 110 degrees in a closed vessel for about an hour. And that should preserve most of the terpenes and things like that in there. That being said, it really actually does depend on what you're hunting for, because all of the different terpenes and cannabinoids will have different temperatures that they volatilize at. And so there are several, if you get online, you can actually look at companies like Leafly and others that will actually have vaporizing guides at what temperature do the various cannabinoids and terpenes volatilize. So that would be a very good resource to answer that question fully for you.
Great. When it does become legal, how would you use it in combination with other herbs, i.e. help with nausea or reduce inflammation? Would it be combined with alkaloid herbs or tannin herbs? Are there no-go combinations, or would it be used on its own?
Yeah, look, that's a really good question. I mean, the first thing that I'd address is that it is legal. It is already legal, and there's about 1,000 patients that have actually gone through clinical trials or special access scheme B, category B, through medical doctors to actually get access. So it is technically currently legal. For us, however, as natural health care practitioners, particularly naturopaths and herbalists, it's not legal to prescribe. And the reason that is because CBD is classified as a Schedule 4, and all of the other cannabinoids, including THC, are basically Schedule 8, which is the same schedule that opiates belong to. So I have given myself the task of, as soon as we actually can get patient access improved and I don't have to lobby so hard to make sure that patients are being taken care of, that will be my next mission, is to actually start lobbying that suitably qualified naturopaths and herbalists should be allowed to access certain components of this. It is a herbal medicine, which I am fairly, I strongly believe that we should be allowed to prescribe this. But in the current climate, I would say that would be several years, and particularly politically, probably several years down the track. However, to answer your second aspect of that question, which was how would it be used? Personally speaking, in my experience, I would use it as a simple. I would use it by itself, much like a lot of herbalists and naturopaths use kava. You kind of use it as rescue medication for, if things get too much. You don't necessarily mix it with other herbs. You use it as a kind of like a pro re nata, as required. And I find that cannabis would probably fit quite well there because it is so phytochemically diverse and can work on so many different mechanisms of action. I would prefer to see cannabis used as a simple, not necessarily because I'm overly concerned about potential herb-drug interactions, just because I think it's an incredibly powerful herbal medicine and probably can carry its own without needing to be mixed extemporaneously with any other herbs. That being said, doesn't mean that it couldn't. That would just be my preference for clinical practice.
Okay, sure. The gentleman that had that question previously that would contact you, he's going to connect with you on LinkedIn, he said.
Fantastic.
Also, we'll just do two more. This is more of an observation from, I've given women with endometriosis a cream with both hemp seed oil and 5% cannabidiol from a GMP pharmaceutical facility. Result was 100% pain loss, N equals 27. That's just something that's I just thought relevant for you to hear. But yeah.
Well, it's super interesting, and that's the thing, is that it's a lot of the anecdotal and case reports that are being that are out there in the population, it's this stuff that we need to share, and particularly from a research point of view, it's very interesting for us because we can take evidence like that and we can potentially formulate and make something a bit more robust and test it. But look, at the end of the day, if that was an N of 27, there's 27 women that have benefited from that. That's really all that matters, isn't it? It's that they've been able to get some symptomatic relief for what is a really nasty and insidious disease. So hats off to you in that regard, and maybe if you're interested, you should be looking at maybe trying to do some research in this space.
I think you're onto it. He's let me know he's a PhD biochemist, so yes, but he'll be connecting with you soon. So
fantastic.
We'll just end on this one. Where can one find a doctor that will prescribe either THC or CBD?
Well, that's a good question because all of the different states and territories actually have different regulations. We used to have a really bad one here in New South Wales, but there's been a great deal of harmonisation between the federal system and the New South Wales system. There used to be duplication of the authorisation process, and it used to take ages and get knocked back, or patients would get approved federally but then get knocked back at the state or the Department of Health level. And so now that's been largely streamlined. So there are a couple of doctors in New South Wales that I'm aware of that are prescribing. There are some clinics as well that are up and running apparently, so cannabis access clinics or something along those lines, where doctors just, I think it's for a rather large fee, which kind of concerns me a little bit. But the reality is that there are popping up. I know of one who is a colleague and patient advocate. Her name is Teresa Taupik. She's a GP up in the Blue Mountains, and I'm familiar that she's helped several patients be able to navigate the special access scheme and get products that are either CBD dominant or THC dominant. But it really actually depends in a way on the condition that you're treating, because if you are trying to manage something, say, that's quite complex like epilepsy, then a GP wouldn't be sufficient to actually prescribe medicinal cannabis for that condition. That would actually need to have the complete support of the pediatric neurologist or just the neurologist in an adult patient. That would actually need to do that. So GPs can write for certain things, particularly pain and other such things, but for more complex conditions, particularly things like multiple sclerosis or, again, neurological conditions, there may need to be specialist involvement. And many specialists, I have a big problem with this because I actually think that GPs are specialists. I think being a GP is far more difficult in a way because you need to take care of every single disease and know every single disease from birth to death. Whereas there are some people out there that just know everything that can go wrong with one particular organ system. So I think that GPs do have a right to prescribe, but the way that it's written in the regulations is that certain specialists, some GPs might actually need to get specialist endorsement to be able to put in a special access scheme category B. But that largely sums it up. I mean, there are quite a few doctors. They're not allowed to advertise. So that's why you probably won't see this in, coming up on television or being in a news magazine type of thing. But the reality is that word of mouth is spreading, and as more doctors are getting trained, and that's something that I've been fairly heavily involved in, is the development of training programs. We actually, through a joint collaboration of the National Institute of Integrative Medicine, the Australasian College of Nutrition and Environmental Medicine, and NICM, Health Research Institute at Western, we put together the medicinal cannabis education course that actually was the first course in Australia that received full Category 1 or 40 CPE points from the Royal Australian College of General Practitioners. So we understand that doctors currently are needed to prescribe this drug, that no one else can, and that it's largely the training of them and the education of them around how to use cannabis that is holding that back. So we're throwing all of our effort into training these healthcare practitioners and medicos so that they feel confident and safe to prescribe this so that we can start getting our patient access numbers up.
Excellent, Justin. I think we'll call it there. There's so many more questions that have come in, and I guess that everyone will just have to follow your previous recordings. Have you got, did you want to tell everyone?
If people are interested in learning a little bit more, you can do one of two things. The first one you can do is check out my website. You can Google me. You'll find it. It's Traditional Medicine Consultancy. You just type in Justin Sinclair Cannabis, it'll pop up. And then if you go to talks and presentations, the tab, generally if I've recorded or had any videos of any of them, the link will be provided for it there. Alternately, what I would actually recommend is probably even better, because you're learning from people that are, leading world experts, is to go to the United in Compassion webpage and click on the Education tab. And in that Education tab will be the recordings and full PowerPoint presentations of all of the major international researchers, academics, and clinicians that we've brought out for our symposiums in 2014, 2016 and 17, I think it was. So we've had 3 symposiums, and we're just getting ready to host our 4th in March next year. So you're learning from some of the world's greatest. So Dr. Jeffrey Hogan-Rather, Dr. Ethan Russo, Dr. Sue Sicily, Dr. Greg Gordon, all of these people that have been able to record these videos, and they're there free of charge for people to educate themselves. So that's what I would recommend. Go and learn from the best. And there's something like 35 one-hour videos there, so all the very best.
Right. Amazing. I just want to, before I do officially sign this off for the night, I just want to say I've just received this in. I've been self-managing my endo for 30 plus years. It is because of this that I initially gravitated to natural medicine, specifically vitamins and minerals, botanical medicine, flower essences, aromatherapy, meditation, breath work. The list goes on. Thank goodness for this research, and thank you. So I think just it's amazing that you're actually focusing on this. So congratulations and seeing the importance of this, and we wish you all the best for your PhD, and we'll definitely follow you from this point onwards if everyone's not already following you. So is there anything else that you wanted to pass on to anybody before we close tonight, Justin?
Oh, look, it's a very thoughtful story, and I love hearing stories from patients. It's how I learn, and it gives me motivation to continue to do what I do. But I did just want to say some thank yous, and firstly, my sincere thanks to you, Raleigh, and the Endeavour team for the opportunity to speak tonight and just share the knowledge. And secondly, thank you to the many, many hundreds of patients that I've met along this journey for me, from infants to centenarians, and too many of whom most regrettably are no longer with us. So I just wanted to thank them for their stories, their time, their constant inspiration, and the countless examples of the power of the human spirit, really. And I'm hoping that we will eventually get there when it comes to this plant, and that we will realize it for the incredible herbal medicine it is, and that hopefully some people listening still might be inspired to seriously consider going down the research route and contributing to knowledge, and maybe doing a master's research or a PhD or honours on cannabis, so that we can actually show the world the evidence that this plant has from 10,000 years of known historical use. So yeah, thank you very much.